Welcome to another installment of the Library Seminar Series, Knauss Fellows 2020. I'm Lisa Clark, one of the reference librarians at the NOAA Central Library. Today's library seminar features two 2020 Knauss Fellows, Lachance and Meredith Richardson. A former Knauss speaker, Caitlin Lauder, International Activities Fellow with the OAR Office of International Activities, will introduce our two speakers, each of whom will speak for 20 minutes, followed by 5 to 7 minutes of questions and answers. But before that, here are a few logistical tips to help you enjoy your presentation today. If you're having trouble with the audio or the visual components of GoToWebinar, please just log out and rejoin the webinar. This usually will resolve our technical issues, but if you're still experiencing problems, let me know by sending an email to library.seminars@noaa.gov. This presentation is also being recorded and will be available on the NOAA Central Library YouTube channel, as well as the Library Seminars website by this afternoon. We'll also be accepting questions throughout the webinar, which the speakers will address at the end of each of their presentations. Please feel free to type your questions in the question box on the right side of your screen at any time. I would suggest, however, distinguishing your questions by adding the speakers' names, Hannah or Meredith, so that we know who your question is aimed at. I thank you for attending this library seminar, and I'm going to turn this presentation over to Caitlin.
Thanks, Lisa. Hannah Lachance received her BS in Animal Science and her MS in Natural Resources from the University of Vermont. She has worked in various areas of research and with various species, spanning from genetics to ecology and moose to fish. Since completing her master's, Hannah has worked for Vermont Fish and Wildlife, USGS Lake Ontario Biological Station, and is currently a 2020 Knauss Marine Policy Fellow at NOAA Fisheries, working as the International Fisheries Science Specialist. Over to you, Hannah.
Hi, everyone. Nice to not see you, but I'm so glad that you're tuning in today to talk a little bit about Cisco, something that I'm very passionate about. Let me just make sure that all of this is going to work. Hold on just a second while I get everything situated here. So Cisco, I'll just start talking, is a great lake species, and you'll probably notice I'm slipping in a couple of puns into my talk because I can't help myself. But generally, they're a native to the Great Lakes, and there's actually a range of species that encompass a group of fish called ciscoes, or you'll sometimes hear me refer to them as corygonids or corygonines. And the singular cisco is generally what I'll use when I refer to this one right here, which is Corygonus artidae. There is other species such as lake whitefish. So if you've been to the Great Lakes, a lot of local restaurants sell lake whitefish, delicious. But if you've heard of lake herring, that's actually referring to this cisco. Some other species I'll be talking about today include kai and hoyi, and these are two deeper water species, and you can tell by their deeper bodies. And generally, they're a unique group of fish. They're actually referred to sometimes as Darwin's finches of the Great Lakes because they adapted to different parts of the lake. They'll feed on different prey within different parts of the water column within different lakes. And because each species fulfills an important ecological role, they're really key in that way because they're connecting different parts of the lower trophic level to the upper trophic level. So something else is that throughout their life, they're an important prey resource. Not only are they fulfilling these important ecological niches, they're fulfilling them at every stage of their life in crucial times of the year. So for example, with cisco, their eggs are generally incubating over winter. So they're an important lipid-rich food source for fish such as lake whitefish to eat throughout the winter. And then their larvae hatch in spring, and they're an important food resource then. And as they continue to grow into adulthood, they're continuously consumed by top predators such as lake trout, salmon species, and muskie. Muskie fishermen actually like to track cisco populations during spawning because they bring the muskie into the bays. So that kind of segues into their economic importance. Since cisco are supporting these ecologically important and economically important species such as lake trout and lake whitefish, which are both sport and commercial fish, they're indirectly supporting the economy in the Great Lakes area. But Cisco themselves are actually becoming a prime source of caviar in Northern Europe. So there's interest in commercially fishing them for that reason. However, due to historical overharvesting and habitat degradation, Cisco and its cousins are nowhere near where they used to be population-wise. And so there's a lot of interest in trying to conserve Cisco, bloater, which is hoyi, one of those deep water species, hai, and others within the Great Lakes. And this interest stems from state agencies, federal agencies, NGOs, universities, Sea Grant, as you can see here. And while each one of them has identified their need for the conservation and restoration, the great thing is that they come together and try to work together to identify research priorities to best address the restoration and conservation efforts needed for each of these species. And so, again, partners such as USGS and US Fish and Wildlife have come together for a workshop on carriganine restoration in all of the Great Lakes, but sometimes there's also lake-specific workshops, such as one organized by a graduate student at Cornell, and with the help of the Nature Conservancy and Sea Grant. And so some of the research priorities that are identified in these workshops include identifying impediments to reestablishment and growth, identifying research gaps in knowledge and life history, genetics and propagation, and understanding the role of genetics and environment on the development of phenotype. And that's where my research starts to come in to try to help address some of these research priorities. And this is a combination of work that I did throughout my graduate time as a master's student at the University of Vermont and throughout my time working with USGS in Lake Ontario before starting my Knauss Fellowship. And the first project I started to tackle in grad school was climate change. Specifically, how does climate change impact Cisco? And then even more specifically, thinking about a crucial time in Cisco life history where climate change could have the most impact, which really is these early life stages, and specifically during winter. Because as I said before, Cisco spawn in late fall, early winter. So right as the ice is starting to come on the lake, the eggs incubate over winter and then hatch right as ice usually melts off in the spring. And so ice seems to be a crucial cue for both spawning and hatching. And unfortunately, while there is variable ice coverage, there is also a significant decline in ice coverage in the Great Lakes, which is predicted to continue. And not only is ice seem to be tied to the spawning and hatching of the eggs, it also seems to be linked with successful recruitment years. So as you can see here on the y-axis, age 1 Cisco density indicates basically a successful recruitment year, the higher it is. And so you can tell the good recruitment years tended to occur when there was a high at maximum annual ice coverage in the Great Lakes. And of course, it's not the key to success in all ways, because there was plenty of years where we had good ice coverage and not good recruitment years, yet it does seem to be important to the good recruitment years in some way, shape, or form. So understanding how declines in ice coverage can impact these species, it can change a couple different environmental factors such as flow, light, and temperature. And temperature has been studied, flows being looked at, but it left this crucial question of how can the change in light resulting from the change in ice coverage impact Cisco. And that's because when there's ice and then snow on top of that ice, it lets only about 7% of the light through. And so Cisco eggs are typically used to incubating in this pretty dark environment. Whereas now, without ice, they can get 100% of that light penetrating down to the depths where their eggs are incubating. So with the help of New York DEC, Tunison Lab of Aquatic Sciences from USGS, and the help of another wonderful graduate student at the University of Vermont named Taylor Stewart, we collected fish from Lake Ontario and fertilized some eggs, brought them back to the University of Vermont, and placed them in one of 3 treatments, either a 24-hour light treatment, regular photo period, or 24-hour dark. And that's because we really wanted to get at if light had any impact, since there were so few studies looking at light and egg development in fish. So Taylor focused on some of the traditional methods, such as measuring their yolk sac volume. So yolk sac being this, I can't quite see what I'm pointing at here because of my face is in my way, but basically both in the egg and the larvae, there's the yolk sac that they feed off of throughout egg development. and larval development. And they need that for the first 2 weeks or so after they hatch to help them switch to exogenous feeding. And so measuring that along with mortality and development rates, we get a better idea of how any light versus dark impacts to the egg, and then the subsequent larval sampling. So to pair with that, I used a molecular method called transcriptomics. And I chose that one because while looking at the genome is interesting to see what genes are there, for this question, we really wanted to know which genes are being used. And to do that, we gathered all the RNA because that's the genes that are being copied and then created into proteins. And so by capturing all the RNA, we can figure out not only which genes are being expressed, but at what level, because we're capturing all of it. So for example, in the light treatment, maybe gene 1 is being used, but it's not being used in the dark treatment. Or, for example, maybe gene 7 is being used at a low level in the dark treatment, but maybe it's being used at a high level in the light treatment. And these kinds of differences stemming from the environment, such as the light, is something important to know in order to try to get at that question of how it can be impacting their survival and growth. And so we sampled both eggs and larvae at 50% hatch, and then we paired it with some of the results from the traditional methods, which basically we saw that the light treatment hatched a full week before the dark and photoperiod treatments. We also saw that the light treatment fish had smaller yolk sacs compared to the photoperiod and dark treatment. And then we also saw in the larvae a higher mortality rate in the light treatment compared to the photoperiod and dark treatment. So keeping that in mind, the transcriptome results, this slide, the main takeaway is that there were genes that were used differently in the eggs and the larvae when comparing the light to the dark treatments. And while there are different genes being used, which is interesting, the main curiosity is what do those genes do? Specifically, what are the ones that are being used differently? And that the main conclusion is that there is increased expression in metabolic pathways. So they're increasing the genes that break down the fatty acids, which kind of makes sense since we were also seeing the smaller yolk sacs. And then, unfortunately, could also explain the increased mortality that we were seeing in the larval population. Now, since this is a pilot study, we can only connect the dots so much, but it definitely merits further investigation because with climate change, people often get caught up in the temperature impacts, which is important. But there are these other environmental cues that also can filter into success or detrimental impacts to the species. So moving on to the next project, we decided to focus on trying to answer some of the life history questions at the larval stage. Because while we have a great diversity of fish in the Great Lakes, specifically ciscoes, and while we can tell them apart visually at this age because of the body depth, the mouth position, etc., at the larval stage, it's pretty much impossible. Once they get a little older, people have better success at trying to tell a few of the species apart, but generally it's still really hard. Also, a few of the species we have successfully reared in captivity, so you can learn a little bit more about the species in that way. But some of them we still haven't, and therefore we're not, really sure of what their typical length at hatch is and what they're eating, etc. And also, if we are capturing a group of fish and we think they're all Cisco, like one species, and we start answering life history questions, and it turns out it's 3 species, then what we thought we knew about the life history is wrong. And also, we really want to be getting at the life history characteristics for each of these species, since each one has such an important ecological role and probably will take a little bit of a different restoration and conservation approach for each species. So the first goal was to see if we could genetically distinguish the larval populations from one another. And then, if we can start answering some of these life history traits. And so to do this, we focused on the Apostle Islands of Lake Superior and sampled from the time the larvae were hatching to the time they disappeared into the juvenile stage. And we sampled 10 sites every single week until that occurred. And at the time, there was a barcoding method. So basically, barcodes, this method used one gene and used an enzyme to cut that gene. and it will cut the gene differently based on the different sequences, which allows you to see different patterns for different species. However, the resolution of this method only let you know if it was a whitefish or not a whitefish, but one of the other chorogonids or ciscoes. And so we still needed to tell the other ciscoes apart. So that was really the group of fish we were focusing on, everything but whitefish, since we can already tell them apart with this method. And existing studies have used the barcoding method and mitochondrial methods, which look at a couple genes, but we wanted to use a hardier sequencing technique to look at as much of the genome as we could justify financially. So we didn't look at the whole genome, but we did look at quite a lot of it, which allowed us to find the little parts of the genes where maybe there are differences between species and therefore be able to tell the species apart. So normally I'd play a fun game with the audience of who's who and let you try to guess the species, but since it's a virtual environment, I will just tell you that we did find 3 different species. And we found Artidae, which is A, and that is Cisco. We also found 2 of the deep water species, Hoyi and Kaiai, which is B and C. And you'll hear me say bloater sometimes. I'm referring to Hoyi in that sense. Kaiai, though, is just always going to be Kaiai. However, we also found a couple of hybrids, which I won't spend a lot of time talking about today. And we also didn't spend as much time investigating because we focused our efforts on the known species. And how we knew that they were these species is, first of all, each dot is a separate larvae. And you can just tell that the specific species clustered pretty well together. However, those would just be clusters, and we wouldn't know which species those actually were unless we compared it to known adults. So luckily, at the same time as we were doing our sequences, collaborators at Wisconsin-Stevens Point, Wes Larson and Amanda Akis, now with NOAA and USGS respectively, were sequencing the genetics of these known adults. So we were able to be confirming that this group of fish was Artidae, Hoyi, and Kaiai. And so now that we know what they are, we can finally start answering some of these life history questions, which is so exciting. So the first one we looked at was where they were and when they were. And generally where they were was, we call it a chorogonid soup. They were kind of appearing all over the Apostle Islands. But when they were was different for each species. So we saw Artidae showing up first. So it generally indicates that they were hatching. And as we move throughout time, you start to see Kaiai appearing. And then you start to see less Artidae because they're starting to grow out into that juvenile stage and leaving the larval populations. But as we move on, you start to see Hoyi appear and Kaiai disappear. So again, Kaiai growing out of the population. So they're showing up at 3 different times. And also this is basically showing the same pattern, but this time we're looking at length. So again, it's just showing that they're smaller when we're first finding them. So they are hatching when we're finding them. And then moving on into the next weeks, we're finding that they grow until they disappear into the juvenile stage. Something else we've looked at was Yolk Sac Condition to kind of keep in mind what we were finding with the climate change project. And what we're finding here is length is on the y-axis. So generally you're seeing, again, they have most of their yolk sac, which is blue, as they're smaller, and then they grow up and absorb that yolk sac. Yet for the 2 deeper water species, we didn't ever see them with their full yolk sac. They generally had partially absorbed or already fully absorbed yolk sacs, which is, interesting, definitely something to ponder a little bit more. But the main conclusion is we can finally tell these larvae apart, which is huge, and it allowed us to dive into some of these other life history characteristics. So again, pairing a suite of techniques allowed us to learn so much more about these species than we have been able to in the past. And then I know we're probably running short on time, but luckily this is a quick project because we're still processing some of the results. But this is some of the work I did with USGS in Lake Ontario. And specifically, we were looking at understanding a little bit more about the spawning habitat that Cisco prefer in Lake Ontario. And so historically in Lake Ontario, Cisco were known to spawn all over the lake, but generally in embayments and generally in near shore, shallow, rocky areas. And in the US, we see that Cisco are only really spawning in this bay right here, Chameau Bay. We found a couple eggs outside the bay, but really that's where we found eggs was within this one bay. In the Canadian waters, there's eggs in the Bay of Quinte over here, but really we'll focus on the US for today. So while we are finding eggs and we're finding them where we typically thought they would be, when we conducted a larval survey in 2018, we were finding larvae, the blue dots, everywhere. And definitely, this is, these are Great Lakes. They're not just a small lake. So too far for the larvae just to drift within the bay, but even outside the bay, et cetera. So that means that cisco are spawning in places that we haven't yet found eggs. And so we need to expand our search, but also we need to maybe reconsider what we thought their spawning preferences were, which led to this project where even within Chameau Bay, we're seeing the eggs, being the orange, were spawned on nearshore shallow areas, but we're finding larvae in areas that have different depths and substrates. So maybe we need to be looking for eggs there too. And that's basically what we did. We thought nearshore shallow, we started looking elsewhere. And so we tried to set traps in a range of areas. However, as beautiful as it is in this video, it's also a challenging time to sample because cisco like to spawn right as the ice is coming on, which makes it hard to get boats out. And so for the sake of trying to learn a little bit more, but also within the constraints of weather, we focused within Chameau Bay since that's where we knew we found eggs before. And here, everywhere that there's red is where we found eggs before. Everywhere that's blue is where we set new traps. And we tried to set across a range of depths and substrates. And generally, we also used what was a traditional mat. So an egg mat, the eggs, it does a great job of collecting the eggs, but it also can collect eggs that drift onto the mat. And since we're really trying to understand where the eggs were spawned and basically where the fish are looking for the habitat, we wanted to capture and retain the eggs. And so we helped design, myself and a bunch of collaborators at USGS and others, modified an existing egg trap called the horns trap into what works better for our purposes of retaining the eggs, preventing predation, and therefore confirming that what we're catching is the number of eggs spawned at that location. And so generally, again, we're still processing these results, and for the sake of time, the main conclusion is that we found eggs almost everywhere we sampled. The highest abundance of eggs still existed in rocky areas that we previously had found eggs, and then a shallow rocky area where we hadn't found eggs before. So that's still pretty interesting that they're spawning elsewhere. And the next question would be to make sure that while they might be choosing these spawning preferences, is it really where the eggs are best surviving? Because as much as they like these shallow rocky areas, it's flat rock. And since these eggs are an important winter subsidy, are they really making it to the spring for hatch? So that would be the next step, is trying to assess the success of hatch from these different substrate types. As I said, this has been a highly collaborative work, and also a work that took a lot of different techniques. And between using the different techniques and collaborating with a wonderful group of people from a wide range of agencies and organizations, we were able to chip away at some of those important research questions outlined to aid in the restoration and conservation of this important native species to the Great Lakes. Without further ado, I'll take any questions that we have at this time, and I'm also happy to take any questions via email or Twitter. And then one final thing is, you probably noticed the hashtag in my title. If you search that hashtag, you get to see a wide range of the work that's being done to study cisco in the Great Lakes. And of course, around winter, it gets a little funky, Disco Inferno. So please check it out. It's a lot of fun.
Awesome. Thank you so much, Hannah. I see a couple of questions. I just want to remind the audience that we're going to take the next 7 minutes to answer them. So please let me send them in. So the first question asks, are there climate change related impacts other than reduced ice cover that may be reducing cisco populations? New predators, change to food web, habitat?
That's a good question. Yes, it's a good answer to that. Obviously, we could spend some time discussing the different options. I would say invasive species is something that, to keep an eye on just for competition of resources. Cisco also really like an oxygen-rich environment. So, you know, if climate change is resulting in maybe a oxygen-depleted environment in perhaps some of the inland lakes a little bit more than the Great Lakes, something to keep an eye on. And then, yeah, there's a couple different areas, but because the early life stage, the eggs and larvae, occurring during that really crucial winter time, and since we think that's where the recruitment bottleneck is, I think that's why a lot of focus is on climate change impacts in the winter for the research around Cisco.
Excellent. This is a compliment. One of our audience members says this was outstanding, so interesting and insightful. Nicely done.
Oh, thank you so much.
Absolutely. One last question is, what is the hashtag to check your research?
The hashtag, I would say Cisco Inferno is probably the one. Sometimes we use like Cisco Inferno 19 for like 2019, etc. But definitely on my Twitter is a lot of the past research, and I like to retweet a lot of the Cisco and Great Lakes stuff. So it's definitely a good hub to stay up to date on some of that stuff.
Definitely adding that hashtag. Okay, well, it looks like we don't have any more questions, but I do want to encourage you to continue asking them. Again, just add Hannah's name to the beginning of the question, because I will send them to her after this presentation to be followed up. So let's go on. I'm going to transfer. Thank you, Hannah, very much. I'm going to transfer the screen now to Meredith.
Great, I'll go ahead and introduce Meredith. Meredith Richardson is a 2020 Sea Grant Knauss Fellow placed in the Office of the Chief Data Officer within the U.S. Department of Commerce. Within this role, she performs two functions: Bureau Liaison to the Chief Data Officer and Executive Secretariat for the Commerce Data Governance Board and NOAA's New Science and Technology Synergy Committee. Meredith is a PhD candidate in the Water Resources Engineering and Science program of the Civil Engineering Department at the University of Illinois at Urbana-Champaign, planning to defend in January 2021. She holds an MS from the University of Illinois and a BS from North Carolina State University, both in civil engineering. In her 4 years at North Carolina State University, Meredith was a Division I scholarship student athlete in volleyball and received a minor in vocal performance.
Can you hear me?
Yep, I can hear you.
Okay, just checking. Thank you so much, Caitlin. Again, Caitlin introduced myself. I'm Meredith Richardson. This is going to be a very different talk than what Hannah just presented. Again, fantastic presentation, Hannah. And so we'll switch gears to more terrestrial ecosystems. First of all, I'm at the University of Illinois, and what you might be wondering is what a person from the University of Illinois might be doing here in a Knauss Fellowship. So I wanted to kind of explain what my curiosity is, and hopefully that'll help you understand more about the context of my PhD. So I wound up in my master's looking at corn, then looking at natural ecosystems for my PhD, and then now in the Knauss Fellowship here. I wanted to point out this is my one trip I got to go on in February at the beginning of my PhD at the Pacific Marine Environmental Lab, and Eugene Berger showed me around there. So this is the sound garden there. But what do all of these things have in common that kind of drive my curiosity? And what they have in common for me is the heterogeneity of Earth systems. What is happening, and why are all these systems the way they are? And so if you look at them separately, you can see that I started with more human managed systems, intensively managed systems, and then I moved to natural terrestrial ecosystems, and now I'm looking at more water-based systems in the Knauss Fellowship. And so this presentation is really going to look at what I'm doing in my PhD. And again, natural terrestrial systems is a big question, so focusing in just on the heterogeneity of vegetation. And then that's still a very big question, because it could be answered on a multitude of scales, from the global scale down to very local scale. And so we are going to focus here on the latter, so very specific sites with particular environmental conditions. And so because of that, now that we know what the context of where we are, we first need to understand what we do know about ecosystems before my PhD started. And so what we know is that there is several quantities or things that an ecosystem must have in order for vegetation to exist. And first of all, it's energy. And so when energy comes into an ecosystem, vegetation will naturally self-organize onto the landscape. And self-organization, what that means is that there's organization that emerges without external predetermination. So that's happening without someone telling it, a tree should go here. So it's a self-organization that happens. And then when this vegetation emerges, then there's also the necessity of water as well in order for it to exist, and nutrients. So those are the three main things that we do know have to exist. But what I would like to know is there's something else occurring within the ecosystem control volume that is helping to determine what vegetation actually exists there. And so that's a little bit more about what this question asks. And so in order to ask this, we have to use our concepts of thermodynamics, open system thermodynamics. And so we start with, you may have heard of closed system thermodynamics, and that's when nothing enters and leaves the system. But here we need to look at open system thermodynamics, where in an ecosystem we have energy coming in and out, and the vegetation is interacting with the surrounding environment. And so that's really important in this context. And then you also have, with every quantity of energy, there's also entropy associated with it. And so the question is, what is entropy? I've gotten many times. Just to keep it simple, entropy, we're talking about thermodynamic entropy here, is the concept of disorder, and which is really the opposite of organization. So if you think of entropy as disorder, low entropy is when there is organization happening. So entropy coming from the sun, that's low entropy, and it is not degraded. It's close to its original source and has more ability to perform work, which I'll get into a little bit later. And then the higher entropy is when there's not much organized and not much organization. It's disordered and highly degraded. And so in an ecosystem with energy going in and out, those are about equal because of conservation of energy. And however, entropy going in is lower than entropy going out. And this is because there's a low local entropy going on within the system. So there's lots of organization. If you look at a leaf, there's the veins within it are very organized. The tree itself is very organized. So this organization that naturally occurs is low local entropy, which leads to an overall higher ecosystem entropy production, which is a jumble of lots of leaves, a jumble of trees, and that sort of thing. So hopefully that gives you a little more understanding of the types of quantities we're discussing here. And so with that in mind and that background, and we think about the energy coming into a system, if we're looking at just a bare soil or an asphalt even, there's a big temperature gradient between the surface and the air. And so that means the temperature surface is very hot and the air is cooler. And that is what we call the temperature gradient. When the temperature difference is over the height. And when vegetation comes into play, when it's naturally self-organized, it simultaneously dissipates the radiation that's incoming, as well as depleting the temperature gradient. And so it dissipates the radiation into several different other energy quantities, such as shortwave, longwave, latent heat, and sensible heat. And however, if you add trees on top of shrubs, for example, that will further cool the temperature surface, or the Earth's surface, and also deplete the temperature gradient further. And so with that leads me to my first research question, does the coexistence of multiple functional groups offer a thermodynamic advantage? And by functional groups, I mean a functional group within an ecosystem is types of vegetation species that perform the same function within the ecosystem. So here we're looking at if trees and shrubs coexist, is that more advantageous than if shrubs exist by themselves or if trees exist by themselves? So that's the question we're asking here. So in order to answer this question, we have to create some more derivative quantities, some other thermodynamic properties. Many of you might have heard the term of work in your physics class. And work, we're going to work, but this is thermodynamic work is a little different in terms of an ecosystem, where we derive it as work is what's considered the energy required to perform motion, to actually move something. And so the work in an ecosystem in terms of vegetation is the latent and sensible heat that is produced, which is in terms of conduction or convection. And then, and so that is really helpful in understanding the ecosystem. However, the shortwave and longwave leaving the ecosystem are radiative energy, and so they're just lost to, they're just heat or energy lost. And so the next term that comes into play helps normalize what's happening in the ecosystem. So it's basically just the work over the incoming radiation. So how an ecosystem performs relative to the energy it's receiving. And so that'll help you understand how efficiently an ecosystem diminishes the temperature gradient that is driving that ecosystem. So now that we understand a lot of these main concepts, I'm not going to get into the modeling framework of what I've been doing. So I use a multi-layer canopy model, which has takes into account the canopy system, the root system, and the soil system. And I'm able to calculate and model the mass, energy, and entropy fluxes in the ecosystem above and below ground. And so this takes in local meteorological data, and this model has been validated time and again on many sites, including the ones discussed here today. And this is, again, a 1D model. And for the specific function, the model was run for different scenarios. So, for example, the different groups are modeled individually, as well as when they are coexisting together. So G1, G2, and then MG, which is the multi-group scenarios when they're modeling coexisting. So here's a brief synopsis of what the results are for this question. What's happening is we are not just showing, we can't add up the results. We are, because we're looking at the dynamics of an ecosystem, we have to look at each of the time steps. And this is, all of this is calculated on a half-hourly time step, and this is over a 2-year period. And on the left, you have the entropy distributions. On the right, you have the work efficiency distributions. And those in blue that I've highlighted here are each of the individual functional groups. So this is the entropy distribution of all the time steps, then each of these, then each of the functional groups separately. And then in red is when they're modeled all together, coexisting and interacting. And so what this means is that a multiple functional group scenario, when they're coexisting, is more efficient at depleting the driving temperature gradient, and it has higher entropy, which more effectively degrades energy. So both of these quantities together help us define thermodynamic advantage. And because they are greater distributions, which were checked with the KS test for significance, as well as the Miller-Jackknife test, they're statistically significantly larger distributions and have larger values. So from what we know now, one other graph I wanted to point out that helps us understand this concept a little more is the work versus temperature gradient plot here in the middle. And so the colored lines here are the different functional groups individually, but the one I want to draw your attention to the most is the black dots in line, and that's showing that there is a power law that exists. So there's a higher power for the multifunctional group scenario than all the other individual group scenarios. And what this means is that there is more work performed for each marginal change in temperature gradient. So that means that the ecosystem is more effective at quickly depleting the temperature gradient, as well as there's more organization that is happening in the ecosystem itself. So that is what that means, and the power law and the importance of that power law and the marginal change. So overall, what these three graphs are showing you is that the coexistence of multiple functional groups is thermodynamically advantageous. So that means in these scenarios that we looked at, an ecosystem will naturally organize towards a multiple functional group scenario with trees and shrubs together, or other types of ecosystems when given the proper nutrients, water, and energy availability. And so from this, it leads me to my question about tree lines. So if that is true, what I just told you, that multiple functional groups is thermodynamically advantageous, then why don't trees exist beyond a tree line? So I show you this picture on the right here because it's a beautiful picture and I love it, but another reason I show it to you is it actually really describes the changes in vegetation and the heterogeneity of the landscape, even within view. So I'm standing here in the foreground on an alpine meadow, and I'm in the Western Italian Alps. And what I'm standing on is an alpine meadow with lots of sedge grass and low shrubbery. And what I'm looking at is the slow migration of the tree line, the alpine tree line. And so what's crazy to me is I'm standing on this grass, this thick grass, and then I see this lower, shorter grasses, and then follow that is the subalpine forest, this evergreen forest. And what I'm wanting to know is what is so different about where I'm standing versus what I'm looking at that is causing this change in vegetation. And so now I'm depicting this in a conceptual diagram, and on the left you have the subalpine site, and on the right you have the alpine site with just the grasses or shrubbery. And again, we understand that ecosystems cannot exist without energy, water, and nutrients. So both of them have those things. They may be slightly altered, even though I can see the difference between the two. Or I can physically see the two. There may be slight differences when you go up in altitude or elevation. But what I want to know is if we look at entropy and work or work efficiency, can that tell us more about why trees don't exist beyond this tree line? So in proper research form, the question I'm asking is, if trees existed beyond a tree line, would they be more thermodynamically advantageous than the existing Arctic tundra or alpine meadow? So I'll delve into this a little more. So I have several sites that I'm going to show you in a second, and each of them, there's the subalpine and subarctic boreal forest or evergreen forest, and then there's the alpine meadow or Arctic tundra. And what I do in this scenario is I construct a counterfactual in which the meadow or tundra has the same vegetation that the subalpine or subarctic forest has. So I simulate the same vegetation as the subarctic forest, but with the same environmental conditions as the alpine meadow or the Arctic tundra, to understand how do the fluxes change. And so there are 3 different pairs of sites with very different climatic conditions. I wanted to include both Arctic tree lines as well as alpine tree lines, so we're not just looking at differences in elevation, but also differences in latitude to see how those changes interplay. And right now we're going to focus only on the Canadian site and the Italy site. So the Italy site is a subalpine evergreen forest, and the site on your right is an alpine meadow, and there are 2.2 kilometers difference in elevation, but they're 19 kilometers difference in distance, so not very far. And the other site we look at is the Tega Plains, Canada. So the one on the left is an Arctic tundra, and you can see that in the winter the vegetation is completely covered. And on the right is a boreal forest, and in the winter the shrubs are covered and the trees are not. So now I'm going to get quickly into the results. So these results are hot off the presses, brand new. I'm still trying to interpret them, but they seem very promising to me, and so I wanted to present them today. Right off the bat, you can see that when the temperature difference is positive, so when the x-axis is positive, we're seeing the same power law that we saw when I was looking at the other sites and the multiple functional group scenario. And we also see that I'm going to point your attention to the colors. So the green is the simulated alpine forest. And then you see that that has the same vegetation as the red, but the same environmental conditions as the blue. And so with this, you can see that the green has less marginal work performed with each increase in temperature difference. And so it has a lower power law, and it seems to be less thermodynamically advantageous from this graph. Another thing to point out is that take a look at the x-axis again. So this is temperature difference in Kelvin. It could also be temperature difference in Celsius. Same thing in terms of difference, but it doesn't really make sense for an ecosystem to have a change in 80-degree temperature from the top of the canopy to the bottom of the canopy. So what's happening is, again, the blue and red figures are validated by the actual ecosystem itself, by in situ observations, but this green, when it's using the same parameters and conditions as those other two sites, is creating an infeasible solution because it's not, because something else is happening within the canopy. And so I think that's an important thing to note. Another thing to note is that when the x-axis is negative, that means there's a negative temperature difference, which means that the temperature of the surface is actually cooler than the temperature of the air of the canopy. And I mention this because this is happening in the blue and the red scenarios, the two real scenarios, but you're not seeing this happen in the green scenario. So I'm going to delve more into that right now. So what's happening in the two real scenarios is what's called a temperature inversion. So what's happening is that the temperature of the air is actually warmer than the temperature of the surface. So in cold regions, this happens. And what's happening is, from the second law of thermodynamics, heat will move from hot to cold. So that means is that the heat is actually moving down into the ecosystem, and the ecosystem is dissipating and capturing it within and below within the canopy and below ground. So that the ecosystem is keeping that heat in and warming itself. However, the simulated forest does not exhibit this phenomenon. It does not have these temperature inversions that exist. It is still having the temperature of the surface hotter than the temperature of the canopy in the air. And because of that, it is resulting in a loss of heat, which is which is not depleting this temperature gradient at all. And so that's why you're seeing those outrageous infeasible numbers in terms of the temperature differences. Oh yeah, and the heat is leaving. So now we've come to the answer of our question. So based on the results that I've presented here today, if trees existed beyond the tree line, it seems that they would not be more thermodynamically a bit advantageous than the existing Arctic tundra or alpine meadow. Again, this is because they perform less marginal work than the other two real scenarios for each temperature difference. And also, they're not exhibiting the temperature inversions that exist in the other sites, the two real sites that would help make it feasible for the plants to perform well. And so with that, I wanted to thank everyone. So I have a lot of figures or a lot of pictures on here. So the university, I wanted to thank my fantastic advisor. He has been so supportive, and our research was funded by NSF. And I'm so thankful to NOAA and the Knauss Fellowship, as well as my host office within the Department of Commerce. So I'm really thankful for that. And again, thank you, fellow fellows. And so last, I wanted to just show you all the different roles I play. I play a lot of different roles in my fellowship, and I'm thankful for that. I get a breadth of experiences. And I wanted to also show you what you would be seeing of me presenting if it were in person, a very active hand gestures, and just show you what it would be like if it were actually in person. So with that, I will take any questions.
No, I think it's great. You did a great job, even without the big hand gestures, Meredith. So yes, it's time for questions. We have, let's see, we have about 8 minutes left in the hour. If anyone would like to send us some questions, you can enter them on the panel on the right side. And I just want to remind everyone that it does take about 30 seconds for it to reach us. But if we miss your question today, not to despair. We do send all the questions to our speakers who then can address them offline. So let's give it another couple of seconds and see if we have any questions. I found this really interesting. I think it's interesting how you went to different locations and different. Oh, here's a question right here. One moment. It says, a great presentation. What do you think was the most rewarding aspect of your research?
Ooh, I want to say going to Italy, but I mean, that was really fun. But really, I think this is going to be a nerdy answer, but I think really learning how to think about questions more clearly and knowing how to get to the answer better. So like the scientific method, learning how to make sure the question that I'm actually asking is the question that I'm addressing. And I think that like clarity of language and just learning how important that is was actually really helpful in this fellowship now. So yeah, that would be just the learning process of the research itself.
Excellent, thank you. There's another question. What was the impetus for your PhD research? Are you building on other research?
Yeah, so as I mentioned, my master's was in corn, and I was looking at corn ecosystems and looking at the nutrient balance there. And at the end of my master's, I was just ready for a change, and I was living in Illinois with the corn, and I wanted to get away from the corn. So I kind of started asking again the big question: why is there heterogeneity on the landscape? And then after talking it through with my advisor a lot, it became clear that the question I was wanting to ask was the heterogeneity of vegetation, and then that led to thermodynamics, which I didn't expect to get involved in, but it was a natural progression. It wasn't laid out ahead of time for me, which I was thankful for. So I got to ask the questions I wanted to ask. Luckily, the model that I mentioned was already developed in our research group. I got to add tweaks to it a little bit here and there and make it relevant for different sites. But the foundation was there, but I could ask the questions I wanted. So
excellent. Another question asks, how do you see this applying to aquatic/marine environments?
Yeah, that's a good question. I'm trying to learn how because this is all based on root uptake and the photosynthesis and how the energy is being depleted or dissipated within an ecosystem. I'd be intrigued to see how that affects when there's like when the water is a medium in between that and the vegetation, or when the vegetation is going through both. So I haven't done that yet, but that would be really interesting to do. But again, I haven't done it, so I don't know what the results would be at all.
Interesting. Another question asks, do the West Coast fires have long-term thermodynamic and environmental impact?
Yeah, that's a good question. That leads me to another part of my PhD that I didn't talk about today, is about direct human impact. So when humans are burning a forest naturally, so if they're doing controlled burns, and then also looking at logging. So that's like direct changes to the structure. So in terms of the California wildfires, that is really going to be directly changing the structure of the ecosystem, which will, again, it'll leave long-term impacts. Without doing the analysis myself, I think it depends on how. I guess I'm just going to say the ecosystem will, I think eventually the ecosystem will return to its original, that gets into the question of critical thresholds and whether an ecosystem is going to go back to its original forest that it was, or whether it'll eventually be catastrophically changed to a different system, maybe even a grassland instead of a forest. And that's a question, another question entirely that I'm trying to get into. But that is a very good question that I'm intrigued to see whether that shift occurs.
Yeah, it is. The wildfires are making us all think a lot. One last question is, fascinating presentation, research, and topic. Can you speak a bit more about your constructed sites and how you developed them?
So like this, I assume it means the simulated sites. So it was not a physical development. It's just all I did was I had the parameters of the vegetation and the parameters of the vegetation and the environmental conditions of the sites, and I modeled them as they exist in reality, separately in those two, so the alpine and then the subalpine. And then when I looked at the alpine site with trees, I used the same environmental conditions that I was using for the alpine meadow, but I just added different vegetation. So I just used the same vegetation from the subalpine site on the alpine site and saw how that changed the fluxes and changed the environmental conditions there.
Excellent. Unless anyone else has a question, I think that is our last question today, and it concludes our hour. Did you have any other comments or questions, either Hannah, Meredith, or Caitlin?
That's all. Thank you.
Thank you. Excellent. Well then, one last reminder that if you joined us late today, or you have a colleague you think might enjoy this Knauss Fellows webinar, please send them the recording. This can be found on the NOAA Central Library YouTube channel or the Library Seminars webpage later this afternoon. I'd like to conclude by thanking our speakers for sharing their research with us, and to Caitlin for introducing both Hannah and Meredith. You guys did a great job today. And to the audience, I really appreciate that you joined us today at the library seminars. I'm speaking for the library when I say that we're very proud to present the work of the NOAA community, and in particular to support the Knauss Fellows. And we hope that you'll all join us again. So be well.
Thank you.